In section F, we begin our discussion of the components of blood with a discussion of plasma. So plasma essentially, of course, is the liquid in which our blood cells travel in the body, but plasma also contains, aside from the formed elements of the blood, some components that are of great interest to us in physiology. The first of those would be the proteins. And so plasma proteins constitute most of the plasma solutes by weight. They play a big role in maintaining blood osmolarity. We talked about the Starling forces of the circulatory system just a moment ago. And these Starling forces, at least the ones that involve osmosis, are mediated mostly by the plasma proteins. So the three main types of plasma proteins are of course the albumins, which make up 55% of plasma proteins and play a big role, as we mentioned, in maintaining the osmolarity of the blood. In other words, drawing liquid into the blood from the surrounding interstitial fluid in the surrounding tissue, and also transport of some, for instance, non-water soluble hormones that need to make their way through the body. The globulins are a second very prominent type of plasma protein that some of which play a major role in immune function, as we are aware, and we'll talk about later in our class. And finally, fibrinogen, which plays a large role in blood clotting, as we'll talk about in just a few moments. So we use the term serum to refer to plasma that has the fibrinogen removed, serum with the clotting factors removed. And so serum is sometimes used in situations like in the case of antivenom, for instance. Serum does contain all the plasma proteins. Well, okay, so we start out with plasma, which contains these three types of proteins. We remove fibrinogen, but we leave albumins and globulins. And so in the case of something like antivenom, we have the globulins, which provide the binding, which conduct the binding to the venom and have the health promoting or life-saving effect here. And so that's a possible use of serum. There are other items found in the plasma as well. There are nutrients that are transported there that are water-soluble, as well as metabolic waste products. Hormones, all the hormones are found in the plasma in one form or another. The definition of a hormone means that they must be transported in the plasma. And we have some electrolytes that we mentioned back in our discussion of bulk flow in and out of capillaries. Those crystalloids, sodium, potassium, and chloride ions are found dissolved in the plasma. We make our way now to blood cells, and briefly we want to mention that all blood cells arise from a type of stem cell known as a pluripotent hemopoietic stem cell. These cells, of course, are found in bone marrow, and they give rise to all the formed elements of the blood, the red blood cells, the white blood cells, and the megakaryocytes, which give rise to the platelets. We focus momentarily on erythrocytes in our discussion here. Erythrocytes, of course, are also known as red blood cells and are responsible primarily for oxygen transport, but some CO2 is transported in red blood cells as well. They contain hemoglobin, and their shape is a biconcave disc, as we see depicted in the lower right-hand corner of our diagram. This shape increases surface area and therefore is adaptive to have the maximum transfer of oxygen and, to a lesser extent, CO2 in and out of the erythrocyte very quickly. Erythrocytes are formed in bone marrow. When they leave bone marrow, making their way into the blood, and we release about 2 million per second, by the way, they leave in a state that's somewhat unusual. An adult or a mature, let's say, a erythrocyte does not have a nucleus. And the immature erythrocyte is referred to as a reticulocyte, and it still contains its nucleus. This reticulocyte exists in that form for about 24 hours before it loses its nucleus and becomes a mature red blood cell. And so reticulocytes are useful diagnostically in some situations because they tell us how quickly red blood cells are being formed. They should constitute about 1% of the circulating red blood cells, but in certain cases, some of which we'll talk about momentarily here, they may constitute more. Erythrocytes don't repair themselves. They have no nucleus and therefore no capacity for repair, and they last about 120 days. This means that about 1% of red blood cells must be destroyed and replaced daily. This is about 250 billion cells. If you break that down, we are apparently releasing 2 million red blood cells every single second into circulation, with, of course, approximately that number of red blood cells removed from circulation, the ones that are worn out at the same time. A discussion of red blood cells involves iron in the body, and so we talked briefly about iron at this particular point in our class. Iron is the element to which oxygen binds in hemoglobin, and so you're aware of the fact that each hemoglobin molecule contains 4 iron atoms, and control of iron, or homeostatic control of iron in the body, would be absolutely essential, is absolutely essential for normal hemoglobin production and therefore normal oxygen distribution in the body. Iron is controlled in the body in the following manner. It's taken up via the intestinal epithelium, and it's not 100% absorbed under normal circumstances, and so there's a great deal of flexibility there to either increase or decrease iron absorption as needed. Iron degraded from hemoglobin is recycled, and so transferrin is a particular protein in the body that returns that iron to bone marrow where it can be reused. This is extremely important because approximately 20 times more iron per day is taken from degraded red blood cells back to bone marrow than is absorbed in the intestinal epithelium. If we look at the overall distribution of iron in the body, we see that it's about half in hemoglobin, half in heme-containing proteins throughout the cells, and half in ferritin. Ferritin is a hepatic iron storage protein that provides some buffer against iron deficiencies in the body. And so we see all these exchanges depicted nicely in our little diagram here. We see that old erythrocytes are broken down by the spleen and liver. That iron is then returned to the bone marrow. Iron circulates from the plasma to all of their cells in the form of these heme-containing proteins, and then, of course, back to the plasma. And then ferritin allows the storage of iron in the liver and serves as a buffer against iron deficiencies. The production of red blood cells is regulated by the hormone erythropoietin. Erythropoietin is produced by some connective tissue cells of the kidneys and moves to the bone marrow where it enhances the production of erythrocytes. Typically, erythropoietin is released in very small amounts, but increased amounts of erythropoietin are produced in response to insufficient oxygen delivery to the kidneys. And so there are multiple reasons for this potential insufficient oxygen delivery. Insufficient pumping of the heart, lung disease, prolonged exercise, or exposure to high altitudes are all possibilities of the reason behind this insufficient oxygen. So as we see in our diagram over to the right-hand side, we have insufficient oxygen delivery to the kidneys, increased erythropoietin secretion, increased plasma erythropoietin, increased production of erythrocytes in bone marrow, increased hemoglobin production, increased oxygen carrying capacity, and then restoration of oxygen to the various tissues of the body. Blood doping is a process of increasing artificially the hematocrit. And so it can be accomplished essentially in one of two ways. One possibility is to withdraw red blood cells, store them, allow the body to restore its original hematocrit, and then add those additional red blood cells back to the body. Another possibility, and a more modern and commonly used one, is the taking of genetically erythropoietin that has been produced by genetically modified bacteria. This is very difficult to detect, and what Lance Armstrong, of course, is now admits he was engaged in. So the additional erythropoietin artificially increases the hematocrit. You can, of course, naturally increase the hematocrit by spending extended periods of time at high altitude, as is done by individuals who are interested in climbing Everest and spend an extended period of time there at base camp at an elevation of over 10,000 feet. We talk next about the other formed elements of the blood, leukocytes and platelets. Leukocytes are, of course, involved in immune defenses of various types. We have 5 subtypes: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. These are covered in more detail in the immune system chapter, so we truncate our discussion here to simply naming them, which is consistent with what our text does. We do mention platelets, though, both their kind of anatomy here and then their function in greater details in the coming slides. Platelets are formed from megakaryocytes. When a megakaryocyte fractures into 1,000 or so smaller pieces, and platelets are odd little creatures in that they can exhibit some sort of movement similar to amoeboid movement, and they secrete a number of compounds that, as you can see, trigger these positive feedback loops, and they're definitely involved in blood clotting. And so we'll talk a great deal over the next few moments about platelets' role in blood clotting. Lastly, we want to mention on this particular slide, in this particular section, that regulation of blood cell production is brought about by so-called hemopoietic growth factors, erythropoietin being the most notable of these. And so the others, interleukins, thrombopoietin, stem cell factor, colony-stimulating factor, the physiology of these is all quite complex, and we don't discuss it in great detail here. What we do notice is that these various hemopoietic growth factors oftentimes have other roles besides regulating blood cell production. They're released by multiple tissues of the body, and in some cases, they prolong the life of the existing blood cells in addition to creating the production of new blood cells. We now, in section 1225, begin a more extended discussion of the prevention of blood loss, which is known as hemostasis. And hemostasis then is most effective in small vessel injuries and in injuries that occur in veins. It's not sufficient to stop the bleeding in most of the arteries that are found in the body, but we do notice that 2 things happen in the case of hemostasis to prevent blood loss. 1, the formation of a platelet plug, and 2, blood clotting. We begin our discussion here with a description of the formation of a platelet plug. The formation of a platelet plug is initiated by the exposure of collagen fibers to the blood. Typically, the negative charges on these collagen fibers are not exposed to the blood. When they become exposed, then Van Willebrand factor, which is a protein that's secreted by endothelial cells and by platelets, adheres to these exposed collagen factors and changes conformation. This leads to the binding of additional platelets to the Van Willebrand factor and the initiation of this platelet plug. So as the platelets bind to collagen, the platelets undergo this so-called platelet release reaction. And these secretory vesicles that are found in platelets release several different components into the immediate surrounding area that bring about this process referred to as platelet activation. The binding in platelets cause them to release these secretory vesicles that contain both mediators such as adenosine diphosphate and serotonin and a compound known as thromboxane A2. Both of these sets of compounds encourage the activation of additional platelets, forming this short positive feedback loop. So if we look at our little diagram here, what we notice is we have vessel damage, we have collagen exposed. The exposed collagen binds to von Willebrand factor, which causes the binding of platelets. That causes the platelets to undergo the release reaction, releasing ADP and serotonin and thromboxane A2. And so these compounds do 2 things. And so they both go ahead and cause the contraction of vascular smooth muscle and vasoconstriction, restricting blood flow to the site. And both of these go back and recruit other platelets to undergo a platelet release reaction. And so over here to the side, we see that eventually this leads to the formation of a platelet plug once we add in the fibrinogen that we'll talk about on the next slide. Here we begin to discuss the role of fibrinogen in the formation of a platelet plug. Fibrinogen receptors on platelets become exposed during this activation process. So the platelets begin to bind to fibrinogen. Imagine fibrinogen as a long protein that's found in the blood. Typically, the receptors for fibrinogen on platelets are not exposed, and so clots don't form. But in response to this injury and the platelet release reaction, the platelet receptors become exposed, and so these long rod-like proteins are binding to platelets, and this promotes the formation of this mesh-like structure with platelets embedded in it. Interestingly, platelets contain a high concentration of myosin and actin, which seems strange, but following this activation reaction, the myosin and actin interact with one another, and so once they are embedded in this plug bound to fibrinogen, then this contraction means that the entire platelet contracts and becomes somewhat more dense, which of course aids in preventing the loss of blood from this particular injury site. Additionally, we see that thromboxane A2 that we mentioned previously is released by platelets and contributes to this platelet recruitment that occurs in the platelet release reaction and this aggregation that takes place. It also acts as a vasoconstrictor. And so that was mentioned in the diagram on the previous slide. But we see that less blood flow is making its way to this area as a result of the release of thromboxane A2. And then lastly, we can ask the question, why doesn't this little positive feedback loop continue to spread throughout the body? In which case, of course, that would lead to death if that clot spread in an uninhibited manner throughout the body. And the answer is the following, that undamaged endothelial cells produce two things that inhibits the spread of this clot. The most important one is prostacyclin, also known as prostaglandin I2 or PGI2. Prostacyclin is a potent inhibitor of the platelet release reaction. And so any platelets that make their way free, any activated platelets that are releasing thromboxane A2 and the other compounds that activate other platelets, those that make their way free should be rapidly inactivated by prostacyclin, because all the healthy, undamaged endothelial cells are releasing prostacyclin. Additionally, as we mentioned previously, those undamaged endothelial cells are releasing nitric oxide. As you know, nitric oxide is a potent vasodilator, and so this is also preventing at least the occlusion of blood flow as a result of a blood clot, since the vessels are increasing in diameter. We now begin our discussion of the role that blood clot formation plays in hemostasis. Remember, blood clot formation is a separate event than platelet plug formation, which we just finished discussing. And so the key here is that we're converting fibrinogen into fibrin. Fibrinogen is a large, as you recall, linear protein that circulates in the plasma, doesn't stick to anything unless platelets have been activated, but when it's converted to fibrin, then it's manufactured into this mesh-like web that we see in the little diagram on our slide. This is a conversion, fibrinogen to fibrin that is, that's mediated by the very important enzyme thrombin. Thrombin is an enzyme that takes several polypeptides, splits them apart in fibrinogen to create fibrin. Fibrin, once you have all these loose fibers, is then combined and strengthened into this web by an enzyme known as factor 8A. And so together, thrombin and factor 8A allow the formation of this mesh-like web of fibrin and the creation of what we know as a blood clot. Various formed elements of the blood, erythrocytes are pictured in this diagram, are trapped in this mesh-like web, and this constitutes our blood clot. So we want to ask the question, how do we initiate the formation of thrombin and therefore the conversion of fibrinogen into fibrin? And so to explain that, we want to go to this diagram that's found in the lower left-hand portion of our screen. It all goes back to this vessel damage, like we talked about in the formation of a platelet plug. And so we have vessel damage, exposure of blood to subendothelial tissue, negative charges, and some other proteins that we'll talk about that are not typically exposed to the plasma. We have an inactivated plasma protein that it becomes transformed into an active enzyme, a second inactive plasma protein converted into an active enzyme, and then this converts the inactive form of thrombin, prothrombin, which is prothrombin, into thrombin. Thrombin then does several things. One thing it does is it actually actively mediates, as we mentioned, the conversion of fibrinogen into fibrin. The second thing it does is it goes to inactive factor 8 and converts it into factor 8A. And the third, 8A then, is taking this loose fibrin, as we mentioned, and turning it into this stabilized web of fibrin by laying in these cross links, like we see there and there and there. The larger structures represent the original thrombin protein, and these smaller segments represent the cross links that are installed by activated factor 8 once it becomes 8A, creating stabilized fibrin. And then thrombin also goes back, as we'll see, and has a positive impact on its own activation. And so we now want to move on and look at these various events that lead to the activation of thrombin and therefore the conversion of fibrinogen into fibrin in more detail. Here we see two independent pathways by which thrombin is activated. One is known, the one on the left is known as the intrinsic pathway. The one on the right is known as the extrinsic pathway. The intrinsic pathway is called the intrinsic pathway because it operates without anything outside of the plasma. The extrinsic pathway requires some proteins outside the plasma to get started. And so what we see is that the intrinsic pathway, for instance, is responsible for blood clotting in vitro, outside the body. For instance, in a test tube. In a test tube, the negative charges in the glass act like the negative charges in collagen and bring about the initiation of blood clotting. So let's go through and talk about the intrinsic pathway first, then the extrinsic pathway, and then how they operate in the body. The intrinsic pathway, then, beginning at this point, if we follow the cursor in our diagram, relies on vessel-damaged and exposed collagen. The negative charges in the collagen expose factor 12 to those negative charges, which activates 12 to 12A. Each of these Roman numerals represents an inactive and an active form of either an enzyme or, in two cases, shown in pink here, a cofactor. So now we have factor 12A, which is an enzyme that converts 11 to 11A. 11A then converts 9 to 9A. 9A is then going to go down and convert 10 to 10A, and 10A is the actual enzyme that's going to convert prothrombin to thrombin. So that's the intrinsic pathway. A couple of different factors that convert, a couple of different enzymes, as we saw in the previous slide, that are activated and eventually lead to the conversion of prothrombin to thrombin, the active form of the enzyme that's actually going to convert fibrinogen into fibrin and lead to the formation of a blood clot. Let's go next to the extrinsic pathway. In the extrinsic pathway, it's different because we have something coming from outside the blood that plays a role. We have vessel damage, we have exposure to subendothelial cells. We have subendothelial cells, that are exposed to the plasma, and those subendothelial cells contain a protein called tissue factor. Tissue factor binds to factor 7, activating factor 7 and turning it into factor 7A. 7A is then going to do two things. It goes down and, just like we saw in the intrinsic pathway, activates factor 9 to 9A, and it actually directly activates 10, turning it into 10A. And so that, of course, gets us back to the activation of prothrombin to thrombin. So either of these pathways can bring about blood clotting because both of them are capable of converting both of them are capable, of converting prothrombin to thrombin and therefore converting fibrinogen into fibrin. What we see in the body, though, is this, that the intrinsic pathway is not potent enough in and of itself to bring about the formation of a blood clot because the factor 12 is not strong enough to fully activate the intrinsic pathway. The intrinsic pathway is very potent, but it can't be activated without the help of the extrinsic pathway because factor 12 is not, there's not enough factor 12 for that to happen. If we go over to the extrinsic pathways, we see that the extrinsic pathway is fairly weak overall for a reason that we'll talk about in a moment. It's largely inhibited by other proteins found in the body. But what the extrinsic pathway does is it typically initiates the formation of a blood clot in the body because it does bring about the production of some thrombin. Once that thrombin is produced, the thrombin goes around and has a positive feedback on its own formation. It converts factor 11 into 11A, which then, of course, cascades down to the production of more 10A and the production of more thrombin. It also activates factor 8 to 8A and 5 to 5A. Both of these are cofactors. The importance of 8A is illustrated by the fact that the most common form of hemophilia is the result of the absence of factor 8, and therefore the nearly complete failure of blood to clot. So to summarize, both of these factors are active in vivo in the body, but they're both necessary for blood to clot because the intrinsic pathway can't get started on its own, and the extrinsic pathway is inhibited to the point where it cannot can't form a blood clot, at least blood clotting that's sufficient to stop blood flow, without some help. So what must happen is the extrinsic pathway initiates a little bit of thrombin. That thrombin initiates the intrinsic pathway, and the thrombin that's produced by the intrinsic pathway is sufficient to form a blood clot. Next, we see that the liver plays some role in blood clotting, and liver diseases are frequently linked to bleeding problems. The liver does two things. One thing that it does is it produces many of the clotting factors that we just discussed. The second thing it does is that it produces bile salts, and so bile salts are released into circulation, and they're necessary for the absorption of vitamin K in the large intestine. Vitamin K is necessary for prothrombin and the production of prothrombin and many other clotting factors, as we see in this little diagram from our text. So the importance of this is illustrated by the fact that some rodenticides, frighteningly, are inhibitors of absorption of vitamin K, which leads to death as a result of excessive bleeding in the rodents. Lastly, we want to talk briefly about factors that oppose the formation of a blood clot. We talked about factors that oppose the formation of a platelet plug, and of course there must also be factors in the body that oppose the formation of a clot, because otherwise that clot would spread throughout the body. We see three. The first is tissue factor pathway inhibitor. And so this is a factor that's secreted by endothelial cells, and it binds to the tissue factor 7A complex and inhibits them from ever making 10A and therefore converting prothrombin to thrombin. This is the main reason that the extrinsic pathway can only make limited thrombin, as we mentioned. It's largely inhibited by tissue factor pathway inhibitor. We also have thrombomodulin, which is a type of cell receptor found on endothelial cells. Thrombomodulin binds to thrombin, and it's expressed all the time on endothelial cells that are exposed to the plasma. And so any activated thrombin that makes its way, thrombin is activated, of course, prothrombin is the precursor. So any thrombin that makes its way away from a blood clot should be bound by thrombomodulin. And then once thrombin is bound, it changes shape and binds to protein C. Once protein C is bound, then interestingly, protein C once becomes activated and inactivates factor 8A and factor 5A, which, as you recall, are both cofactors. Factor 8A is the cofactor that, when it's absent, leads to the most common form of hemophilia. So thrombomodulin does two things. It gets rid of thrombin, and it inactivates these two cofactors that are necessary for the production of more thrombin. Lastly, this is not pictured here, but we have a third factor, antithrombin 3, which is a plasma protein that binds to thrombin and inactivates thrombin and other clotting factors. Its activity is enhanced by heparin. You're familiar with the role of heparin as an anti-clotting agent. If you're taking blood in the laboratory, you may place it into heparinized tubes if you want to prevent it from clotting. And so this anti-clotting activity is mediated through antithrombin 3. We'll omit the fibrinolytic system found on page 438, and this will bring our discussion of Chapter 12 to a close.